In 1938, a museum curator in South Africa found a strange blue fish among a fisherman's catch, a species scientists had confidently declared extinct for 66 million years, and it forced an entire branch of biology to admit it had simply been wrong. That's tonight's first fact, and it's only the beginning of 180 more, all new, all about the same vast dark world beneath the waves, none of them repeated from this channel's earlier deep ocean video. We'll go looking for underwater lakes that sit at the bottom of the ocean without ever mixing into the water around them, sharks that may live for 4 centuries and only reach adulthood around age 150. A mysterious sound recorded in 1997 that took scientists 8 years to explain, and an octopus that guarded her eggs for nearly 5 years without eating once. We'll walk through a landscape stranger and flatter than almost anyone pictures, and end with the slow, patient instruments now watching the deep sea change in real time. Some of tonight's mysteries have been carefully solved. A genuine number of them haven't. Let the water close slowly over you, and let the deep carry you down. Long before anyone can discuss the creatures living on the deep ocean floor, it helps to understand what that floor actually looks like. And the honest answer, for most of it, is considerably stranger and flatter than most people imagine. The single most common landform on the entire surface of planet Earth is not a mountain, a plain, or a desert, but abyssal hills, low, gently rolling undulations rising just tens to a few hundred meters above the surrounding seafloor, formed as tectonic plates pull apart at mid-ocean ridges, and the newly created crust fractures and cools unevenly as it spreads outward. A comprehensive 2014 global seafloor survey led by researchers including Goff and Arbic, compiling decades of ship-based sonar data, confirmed that abyssal hills cover a genuinely staggering proportion of the entire ocean floor, considerably more surface area than any single terrestrial landform type covers on land. And yet the feature remains almost completely unknown outside oceanographic circles, specifically because it lies, on average, several kilometers beneath the surface, entirely invisible to anyone who has never seen a detailed seafloor map. Between these hills stretch the abyssal plains themselves, some of the flattest, natural surfaces found anywhere on Earth, with slopes uncertain. Regions measured at less than 1 meter of elevation change per kilometer of horizontal distance, a flatness achieved through a slow, patient geological process, a continuous rain of fine sediment called marine snow settling from the productive surface waters far above, gradually burying the rougher volcanic terrain beneath a smooth blanket that can, in the oldest and most sediment-rich regions, exceed a kilometer in accumulated thickness. This sediment itself comes in genuinely distinct types depending on location and depth, and oceanographers classify much of it as ooze, a technical term rather than a casual description, referring specifically to sediment composed of at least 30% the skeletal remains of tiny marine organisms. Calcareous ooze, built from the calcium carbonate shells of foraminifera and coccolithophores, dominates shallower abyssal regions, while siliceous ooze, built from the glassy silica skeletons of diatoms and radiolarians, accumulates in specific high-productivity zones, including the waters surrounding Antarctica, and red clay, the finest and slowest accumulating sediment type of all, composed largely of wind-blown dust and volcanic ash that has drifted down through the water column over such enormous spans of time that it dominates only the very deepest, most biologically unproductive ocean basins, where dissolving calcium carbonate never even reaches the bottom intact. Cutting directly through this otherwise gentle landscape are submarine canyons, dramatic gorges that can rival or exceed the scale of the largest canyons found on land, carved not by rivers but by turbidity currents, fast-moving underwater avalanches of sediment and water that can travel for hundreds of kilometers along the seafloor at speeds exceeding 20 meters per second once triggered, typically by an earthquake, a storm-driven sediment collapse near a river mouth, or a slope failure on the upper continental shelf. Monterey Canyon, off the coast of central California, illustrates the scale involved directly, plunging more than 1,000 meters deep and stretching over 150 kilometers, comparable in scale to Arizona's Grand Canyon, despite sitting entirely underwater, and remaining, for that reason, almost completely unknown to the millions of people who visit the nearby coastline every year without ever suspecting what lies just offshore. Researchers are monitoring Monterey Canyon with permanently installed seafloor instruments have directly recorded active turbidity current events multiple times since sustained monitoring began in the 2000s, including a well-documented January 2016 event that a research team led by Woods Hole geologist Catherine Meyer confirmed traveled through the canyon at sustained speeds exceeding 8 meters per second, carrying enough sediment to measurably reshape sections of the canyon floor within a matter of hours. Geological change on a timescale usually associated with a single dramatic storm rather than the slow, patient pace most people associate with the deep sea. At the opposite geological extreme from the ocean's gently rolling abyssal hills sit the deep Haddle Trenches discussed extensively in this channel's earlier deep ocean video, and their distinctive narrow, V-shaped profile, dramatically different from the broad, gradual slopes found almost everywhere else on the seafloor, results directly from the specific mechanics of plate subduction, the process by which one tectonic plate is forced downward beneath a neighboring plate at a convergent boundary. As the denser, typically oceanic plate bends and plunges beneath the adjacent plate, it drags the immediately surrounding seafloor downward with it before the two plates make full frictional contact at depth, creating a narrow, steep-sided depression considerably more confined in width than its extreme depth would otherwise suggest. A specific structural signature geologists can use to identify a subduction zone from bathymetric data alone, distinguishing it immediately from the broader, gentler basins and gradual continental slopes that dominate the rest of the seafloor's overall shape. Rising abruptly from these otherwise gentle plains and canyon systems are the mid-ocean ridges, the longest continuous mountain chain on the entire planet, stretching for more than 65,000 kilometers across every major ocean basin on Earth, a connected volcanic system considerably longer than the combined length of every mountain range found on land. The ridges mark the boundary where tectonic plates are actively pulling apart, with new oceanic crust continuously forming as magma rises to fill the gap, a process oceanographers only confirmed through direct visual observation and precise measurement, beginning in the 1970s when specially equipped research submersibles first allowed scientists to directly witness active seafloor spreading rather than inferring it purely from indirect magnetic and sonar evidence gathered from the surface. Despite representing, by total surface area, one of the single largest continuous geological features on the entire planet, the mid-ocean ridge system remains so poorly mapped in fine detail that oceanographers routinely note humanity has produced considerably more detailed topographic maps of the surface of Venus, an entirely separate planet obscured by thick clouds and mapped only through orbital radar, than of Earth's own primary mountain-building system, sitting directly beneath the ocean that covers most of the planet we actually live on. Before we go further, if slow, careful descents like this one are the kind of thing that helps your mind finally unwind at the end of a long day, this channel makes a habit of building them, and subscribing costs nothing beyond making sure the next one finds you. Now, back to the landscape itself. A final, genuinely counterintuitive feature of the deep ocean floor deserves mention specifically because it upends most people's basic intuition about how sediment accumulates in the deep sea. The carbonate compensation depth, a specific boundary, generally sitting somewhere between 4,000 and 5,000 meters, depending on location and ocean chemistry, below which the water becomes cold and pressurized enough that calcium carbonate, the mineral making up the shells of countless tiny marine organisms, actually dissolves faster than it can accumulate on the seafloor. Above this boundary, the seafloor is frequently blanketed in the pale calcareous ooze described earlier, built from the slow accumulation of undissolved shells. Cross below it, and that same falling shell material simply dissolves away entirely before ever settling permanently into the sediment, leaving behind only the slower accumulating silica-based or clay sediments that survive the deeper water's more corrosive chemistry, meaning the seafloor's basic sediment type changes dramatically and predictably with depth alone, a boundary invisible to any observer at the surface but immediately obvious to anyone examining sediment cores pulled from different depths within the same ocean basin. Marine snow itself, the continuous gentle downward drift of organic particles that ultimately builds so much of the abyssal plain's sediment cover, deserves a closer look before this chapter moves on, since the term describes something considerably more varied than a simple, uniform rain of debris. Researchers studying marine snow composition, directly, primarily through specialized underwater cameras and sediment traps moored at various depths, have found the falling material includes dead phytoplankton, fecal pellets from surface-dwelling zooplankton, discarded mucus-feeding structures shed by small gelatinous animals, and clumps of bacteria, all bound loosely together into aggregates that can range from barely visible specks to particles several centimeters across, some large and cohesive enough to be individually tracked descending through the water column on camera. A single marine snow particle can take anywhere from several days to several weeks to complete its full descent from the sunlit surface to the deep ocean floor, depending on its size, density, and the specific currents it encounters along the way, and researchers calculating the overall rate of this downward flux have found it varies considerably by region and season, generally peaking following surface phytoplankton blooms, meaning the deep sea floor experiences its own distinct delayed echo of surface productivity cycles occurring in sunlit water that may sit several kilometers and several weeks removed from the particles that eventually settle there. The mid-ocean ridge system discussed above provided the physical setting for one of the 20th century's most elegant pieces of geological detective work published in 1963 by British geophysicists Fred Vine and Drummond Matthews, building on a closely related and independently developed idea proposed around the same time by Canadian geologist Lawrence Morley. Vine and Matthews, analyzing magnetic surveys of the seafloor surrounding mid-ocean ridges, noticed the rock formed a striking pattern of alternating magnetic stripes running roughly parallel to the ridge itself, symmetrical on either side, and proposed a genuinely elegant explanation. As new oceanic crust forms continuously at the ridge and slowly spreads outward in both directions, it cools and locks in the orientation of Earth's magnetic field at the exact moment of its formation, and because Earth's magnetic field is known from independent geological evidence to periodically reverse its polarity entirely over timescales of hundreds of thousands to millions of years, each new band of crust records whichever polarity happened to be in effect when it solidified, producing a continuous symmetrical magnetic Kirchhoff stretching outward from the ridge on both sides like two matching strips of recording tape pulled apart from a single source. This magnetic striping evidence, combined directly with Harry Hess's Guyot-based seafloor spreading proposal, provided the final, conclusive physical confirmation needed to establish plate tectonics as settled scientific consensus by the late 1960s, and researchers can now use the width of individual magnetic stripes, combined with the independently established dates of Earth's past magnetic reversals, to calculate the exact historical rate of seafloor spreading at any given ridge segment with genuine precision, a technique that remains one of the primary tools geologists use to reconstruct the ocean floor's geological history today. Long-term sediment cores drilled from the deep seafloor gathered through decades of coordinated international scientific drilling programs, beginning with the Deep-Sea Drilling Project in 1968 and continuing through its later successor programs, have provided researchers with a genuinely remarkable secondary benefit beyond simply documenting sediment type, a continuous layer-by-layer climate record stretching back tens of millions of years, since the specific ratio of oxygen isotopes preserved within the calcium carbonate shells of foraminifera accumulating in seafloor ooze varies measurably with the ocean temperature and global ice volume present at the time each shell layer originally formed. Researchers analyzing these deep-sea sediment cores have reconstructed a detailed record of Earth's ice age cycles stretching back more than 2 million years, confirming the regular, roughly 100,000-year rhythm of major glacial and interglacial periods first proposed on theoretical grounds by Serbian astronomer Milutin Milankovic in the early 20th century, connected to slow, predictable variations in Earth's orbital shape and axial tilt, meaning the same slow-settling ooze covering the abyssal plain that most people would assume holds no information at all, has instead quietly preserved one of the most detailed and scientifically important climate archives available anywhere on the planet. The single largest waterfall on the entire planet, measured by the sheer volume of water in motion, is not a feature any person could ever stand by it and observe directly, since it exists entirely underwater, submerged within the Denmark Strait between Greenland and Iceland. The Denmark Strait cataract forms where a considerably denser body of cold Arctic water, flowing south, encounters and abruptly plunges beneath the comparatively warmer, less dense water of the Irminger Sea, cascading downward across a submerged drop of more than 3,500 meters, a vertical descent well over twice the height of Africa's Angel Falls, the tallest waterfall found on dry land, and moving an estimated volume of roughly 5 million cubic meters of water per second, a flow rate oceanographers calculate as many times greater than the combined volume of every river on Earth flowing into the ocean at once. The cataract was first mapped in detail only through modern satellite altimetry and sonar surveys conducted in the 1990s, since its entirely submerged nature made it invisible to centuries of surface-based nautical observation, and researchers studying the feature consider it a genuinely significant driver of the broader thermohaline circulation system discussed later in tonight's video, since the volume and density of cold water plunging through the strait directly influences how much cold, oxygen-rich water ultimately reaches the deep Atlantic Ocean floor further south. For most of the 19th century, one of the most confidently stated scientific conclusions about the ocean turned out to be flatly wrong, and the story of how that error was corrected, decades before any submersible existed capable of directly checking the claim, offers a genuinely instructive window into how deep-sea science actually developed long before the modern exploration era most people picture when they think about ocean discovery. British naturalist Edward Forbes, one of the most respected marine biologists of his era, proposed what became known as the Azoic hypothesis in 1843, based largely on his own extensive dredging surveys of the Aegean Sea, concluding that animal life became progressively sparser with increasing depth and effectively disappeared entirely below roughly 550 meters, a boundary he termed the Azoic zone, reasoning that the combination of crushing pressure, total darkness, and near-freezing temperature would make survival biologically impossible past that point. Forbes's hypothesis, delivered with the full institutional authority of a leading Victorian scientist, was widely accepted throughout the scientific community for decades, afterward shaping how an entire generation of naturalists and even early ocean engineers thought about the fundamental limits of life on Earth, despite resting on a genuinely limited sampling depth by later standards, and despite Forbes himself dying in 1854 well before the evidence that would ultimately overturn his conclusion had fully accumulated. That overturning evidence arrived almost accidentally through an entirely unrelated engineering project: the laying of transatlantic telegraph cables across the floor of the North Atlantic, beginning with an initial short-lived success in 1858 and culminating in a permanently functioning connection in 1866, engineered under the direction of British physicist and engineer William Thomson, later ennobled as Lord Kelvin specifically in recognition of his contributions to the project. When damaged sections of early telegraph cable were periodically hauled back up from the seafloor for repair and inspection, engineers and the naturalists who examined the recovered material found the cables thoroughly encrusted with corals, mollusks, and other clearly living marine organisms retrieved from depths considerably greater than Forbes's proposed azoic boundary. Direct physical evidence, recovered as an incidental byproduct of a communications engineering project rather than any dedicated biological expedition, that life persisted at depths Forbes had confidently declared impossible. This cable-derived evidence, considerably more than any single dramatic counterexample, helped build the case that ultimately led the British Admiralty and Royal Society to authorize the voyage of HMS Challenger in 1872, and naturalist Charles Wyville Thomson, who served as the Challenger expedition's chief scientist and had himself argued publicly against Forbes's azoic hypothesis years before the voyage began, made confirming or refuting the depth limit on animal life one of the expedition's explicit primary scientific goals. Challenger's dredging operations, conducted at stations scattered across every major ocean basin the ship visited during its 4-year global circumnavigation, recovered living animal specimens from every single depth sampled, including stations exceeding 5,000 meters, definitively and comprehensively disproving the azoic hypothesis by the expedition's conclusion and establishing, for the first time with rigorous, systematically gathered evidence, that some form of animal life extends essentially to the deepest points of the ocean, a foundational finding of modern deep-sea biology achieved entirely without any vehicle capable of actually carrying a human being down to see that life directly. Direct human observation of deep-sea life, as opposed to indirect study through dredged and trawled specimens, would not arrive for several more decades, and when it finally did, it came through a strikingly simple piece of engineering: the bathysphere, a spherical steel diving chamber with no propulsion system of its own at all, designed by engineer Otis Barton and lowered on a steel cable directly from a surface support ship first used for a series of pioneering dives off the coast of Bermuda, beginning in 1930 by naturalist William Beebe, who occupied the cramped sphere alongside Barton on numerous descents and provided a running verbal account of what he observed, relayed back to the surface ship through a telephone cable bundled alongside the sphere's suspension cable. On August 15, 1934, Beebe and Barton reached a depth of 923 meters, becoming the first human beings in history to directly observe with their own eyes the deep sea's total darkness and its resident bioluminescent life, a milestone considerably predating the bathyscaphe Trieste's famous 1960 descent to the Challenger Deep by more than a quarter of a century, though Beebe himself candidly acknowledged in his own popular writing that several of the specific unfamiliar creatures he described peering out through the bathysphere's small quartz windows during these dives, given only fleeting glimpses and possibility of specimen collection, were never independently confirmed by later expeditions, leaving a genuinely open historical question about exactly how reliably some of Beebe's more startling verbal descriptions actually matched what he saw, versus what excited pattern-seeking under difficult observing conditions may have led him to interpret. The bathysphere itself carried a genuinely severe limitation that shaped and ultimately ended its usefulness for serious ocean exploration. Lacking any independent means of propulsion, buoyancy control, or emergency ascent capability, it remained entirely dependent for its entire operational life on the mechanical integrity of a single steel cable and winch system aboard the surface support ship, meaning any failure of that cable, or of the ship's ability to maintain station directly above the sphere in rough seas, would leave its occupants with no means of self-rescue whatsoever, a risk profile considerably more precarious than the self-contained bathyscaphe design Auguste Piccard would go on to develop specifically to address this exact vulnerability barely more than a decade later, incorporating the sphere's occupied chamber into a larger, independently buoyant vessel capable of controlled ascent and descent without depending on any surface cable at all. An earlier and considerably less celebrated figure, did more than perhaps any other single individual to professionalize deep-sea research in the decades between Challenger's pioneering 1870s voyage and Beebe's bathysphere dives, and he happened to be reigning European royalty. Prince Albert I of Monaco, who ruled the principality from 1889 to 1922, personally funded and led a series of oceanographic expeditions aboard 4 successive purpose-built research vessels, all named Hirondelle or Princess Alice, conducting deep-sea trawling and specimen collection across the North Atlantic beginning in the 1880s and continuing for more than 3 decades, work that included some of the earliest systematic studies of deep-sea squid and whale biology conducted anywhere in the world. Albert founded the Oceanographic Museum of Monaco, which opened in 1910 and remains an active research institution today, specifically to house and display the specimens and findings his own expeditions had gathered, and he separately established the Oceanographic Institute in Paris the same year, creating, through his personal wealth and sustained scientific commitment, one of the first dedicated institutional homes for oceanography as a formal academic discipline anywhere in the world, decades before most national governments treated ocean science as a field worth funding at any comparable scale, a genuinely significant contribution to the field's early professional development that receives considerably less popular recognition today than the more dramatic vehicle-based milestones that followed it. French naval officer and ocean explorer Jacques Cousteau, already widely known by the late 1950s for co-inventing the aqua lung, the self-contained underwater breathing apparatus that made recreational and scientific scuba diving practical, pursued a genuinely different approach to deep-sea access than either the bathysphere or the bathyscaphe, developing a small maneuverable 2-person submersible called the SP-350. Denise, nicknamed the diving saucer for its distinctive rounded shape, first tested in 1959, and capable of reaching depths of around 350 meters under its own independent power, considerably shallower than Trieste's record-setting Mariana Trench dive, but far more agile and reusable for routine scientific work along continental slopes and canyon walls than either of the earlier, far more cumbersome designs. Cousteau pursued an even more ambitious, and in hindsight, considerably riskier line of research during the same general period, called the Conshelf experiments, conducted between 1962 and 1965, in which small teams of aquanauts lived for extended periods, up to nearly a month during the final Conshelf 3 experiment, inside pressurized underwater habitats positioned directly on the seafloor at depths reaching about 100 meters, breathing specially formulated gas mixtures and venturing out on working dives directly from their submerged living quarters rather than returning to the surface between excursions. The Conshelf program demonstrated that sustained human habitation at meaningful ocean depth was technically achievable, and it directly influenced later underwater habitat programs, including the U.S. Navy's Sela B project and NASA's present-day NEMO underwater astronaut training program, though the genuinely serious physiological risks involved in prolonged saturation diving at depth including decompression complications discovered and refined only through this exact era of experimental, occasionally dangerous human trial, ultimately limited how far the underwater habitat concept was ever practically extended beyond these pioneering mid-century experiments. The deep ocean's extreme stability, its unchanging cold, its permanent darkness, its consistent pressure, has made it an unusually effective refuge for lineages that have otherwise vanished everywhere else on Earth, and few discoveries illustrate this more dramatically than the Coelacanth, a fish belonging to a lineage scientists had confidently declared extinct for approximately 66 million years, believing the entire group had died out alongside the non-avian dinosaurs at the end of the Cretaceous period. On December 22, 1938, museum curator Marjorie Courtenay-Latimer, working at a small natural history museum in East London, South Africa, was called to examine an unusual catch brought in by a local fishing trawler operating off the mouth of the Shalumna River, and among the ordinary fish sat a large, steel-blue specimen unlike anything she had ever encountered, with distinctive fleshy, lobed fins rather than the thin, ray-supported fins of ordinary bony fish. Courtenay-Latimer preserved the specimen as best she could given the region's limited facilities, and sent a detailed sketch to South African ichthyologist J.L.B. Smith, who, upon finally examining the preserved specimen in person several months later, immediately recognized it as a living coelacanth, a discovery he later described as feeling, in his own words, as though he had encountered a living dinosaur walking down the street, and one that was formally published in the journal Nature in 1939, sending immediate shockwaves through the international paleontological community. The species was formally named Latimeria chalumnae, honoring both Courtenay-Latimer's role in its discovery and the river near which it was caught, and its rediscovery is now widely regarded as one of the single most significant zoological findings of the entire 20th century, precisely because it demonstrated, with total and undeniable certainty, that an entire vertebrate lineage confidently pronounced extinct by the fossil record could in fact still be quietly persisting, entirely undetected by science, in a specific, poorly surveyed deep-water habitat. A second, genetically distinct Coelacanth population was subsequently discovered off the coast of Indonesia in 1997, spotted initially by traveling biologist Mark Erdman while on his honeymoon, who noticed an unusual fish being sold at a local market and recognized it immediately from photographs of the African species, a population later confirmed through genetic analysis to represent a fully separate species, Latimeria menadoensis, having diverged from its African relative an estimated 30 to 40 million years ago, meaning two independent coelacanth lineages had each separately survived, entirely undetected by modern science, in two widely separated ocean regions. Coelacanths are now understood to inhabit deep underwater caves and steep reef slopes, generally at depths between 150 and 700 meters, considerably shallower than the true abyssal depths discussed elsewhere in tonight's video, but still well beyond the range of ordinary recreational diving, and researchers tracking tagged individuals have found coelacanths can live for remarkably long periods, with a 2021 study analyzing growth rings in coelacanth scales, led by French researcher Kayleigh Mahé, proposing the species may live to nearly a century old, growing and reproducing at a pace so extraordinarily slow that female coelacanths may not reach reproductive maturity until roughly 55 years of age, an estimate that, if accurate, would make coelacanths among the slowest maturing vertebrates ever documented, and one that carries serious conservation implications, given how vulnerable such a slow-reproducing species would be to even modest additional fishing pressure. A second genuinely striking living fossil inhabiting deep continental slope waters is the frilled shark, Chlamydocelicus anguineus, a species whose eel-like body, unusual gill structure, and distinctive backward curving Needle-like teeth arranged in rows have led researchers to describe it as retaining a body plan more closely resembling ancient shark ancestors from the fossil record, some dating back more than 80 million years than nearly any other shark species alive today. Frilled sharks typically inhabit depths between 500 and 1,000 meters and are almost never observed alive, given how rarely they venture anywhere near the surface, though a rare and widely reported January 2007 encounter off the coast of Japan, in which a frilled shark was captured alive, apparently disoriented and in poor health, near the surface and briefly filmed at Awashima Marine Park before dying, offered one of science's few direct video glimpses of the living species, and researchers studying the specimen afterward proposed it had likely become disoriented after venturing into unusually shallow water, possibly while sick or otherwise struggling, since healthy frilled sharks are essentially never encountered anywhere close to the surface under ordinary conditions. A third deep-water lineage frequently discussed in the same breath as these living fossils, the goblin shark, Mitsucarina austoni, named for the distinctly elongated, flattened snout extending well beyond its jaw, represents the sole surviving member of an entire shark family, Mitsucarinidae, whose fossil lineage stretches back more than 125 million years, with every other known genus within the family long since extinct. Goblin sharks possess a genuinely remarkable predatory adaptation unmatched by nearly any other shark species, a set of specialized jaw ligaments allowing the jaw itself to rapidly extend forward, snapping shut around prey. In a motion, researchers studying filmed specimens have compared to a slingshot, a striking mechanism first captured on video only in the 2000s after decades in which the species was known almost entirely from trawl-caught, already deceased specimens, since goblin sharks, inhabiting depths generally between 250 and 1,300 meters, are only very rarely encountered alive by researchers at all. Stalked crinoids, or sea lilies, an ancient group of echinoderms, the same broad animal group that includes starfish and sea urchins, offer a further, less individually dramatic, but geologically far more extensive example of the same deep-sea preservation pattern. Crinoids were extraordinarily abundant throughout Earth's shallow seas during the Paleozoic and Mesozoic eras, so abundant that entire, thick layers of sedimentary limestone found across the modern world are composed almost entirely of their fossilized skeletal plates. Yet the group appeared to decline dramatically and nearly vanish from the fossil record following major shifts in shallow marine ecosystems, leading many 19th-century paleontologists to treat stalked crinoids as a group in its final stages of natural extinction. Deep-sea dredging expeditions, beginning with the same HMS Challenger voyage discussed earlier in tonight's video, and continuing through later 20th century submersible research, instead found stalked crinoids thriving in genuine abundance at depths generally below 200 meters, anchored to the seafloor by a long, flexible stalk and filter feeding, with feathery arms extended into passing currents, revealing that the group had not gone extinct at all, but had instead, across tens of millions of years, gradually retreated into deeper water as shallow water predation pressure from newer fish and crab lineages intensified, a genuinely instructive case in which an entire major animal group essentially relocated its center of survival to the deep sea, rather than actually dying out, hiding in plain sight from paleontologists who had only ever studied its shallow water fossil record. The chambered nautilus, Nautilus pompilius, and its several close relatives, represents the last surviving remnant of externally shelled cephalopods, a body plan that once dominated the ancient oceans in extraordinary diversity, including the ammonites familiar from countless fossil collections, before nearly the entire external shell cephalopod lineage vanished during the mass extinction event that ended the Cretaceous period. Alongside the non-avian dinosaurs roughly 66 million years ago, leaving the Nautilus genus as essentially the sole surviving branch of a once vast evolutionary tree. Unlike squid and octopuses, its considerably more familiar internal shell or shell-less cephalopod relatives, nautiluses retain a coiled external shell divided internally into a series of sealed gas-filled chambers, and researchers studying nautilus buoyancy control have found the animal actively regulates the fluid and gas balance within these chambers to precisely manage its own depth, functioning as a genuinely sophisticated biological submarine ballast system considerably older than any comparable human engineering by many tens of millions of years. Nautiluses undertake their own daily vertical migration remarkably similar in basic structure to the twilight zone migration discussed elsewhere in tonight's video, generally resting at depths of 300 to 500 meters or more during the day to avoid predators, and rising to considerably shallower reef slopes at night, specifically to feed. And researchers have found the species' external shell can withstand pressure depths down to roughly 800 meters before the structure begins to fail, a hard physical depth limit that appears to define the outer boundary of the species' entire vertical range, a genuinely ancient body plan still quietly performing the same basic daily commute its extinct ammonite relatives most likely also once made, long before anything resembling a modern fish had evolved to share the same water with it. A fourth living fossil, considerably less famous than the coelacanth, but arguably even more startling to the specialists who first encountered it, is Neopelina galathi, a small, limpet-like mollusk belonging to a class called Monoplacophora, a group paleontologists had confidently assumed extinct for roughly 375 million years, known previously only from fossil shells dating back to the early Paleozoic era. Danish researchers aboard the Galathea expedition dredged the first living specimens from a depth of roughly 3,570 meters off the coast of Mexico in 1952, and formal scientific description of the find, published in 1957, revealed a genuinely significant anatomical surprise beyond the animal's mere survival. Neopelina's internal organs, including its muscles, gills, and excretory structures, are arranged in a distinctly segmented, repeating pattern along the length of its body, a body plan that helped settle a long-running debate among zoologists about the evolutionary relationship between mollusks and segmented animals like earthworms and arthropods, since Neopelina's living anatomy offered direct physical evidence that the mollusk lineated from a more segmented ancestral body plan than modern snails, clams, and octopuses, none of which show any comparable segmentation, had previously suggested. Additional Monoplacophora species have since been discovered at various deep-sea locations worldwide, and researchers studying the group generally regard it, alongside the Coelacanth, as one of the clearest demonstrations that the deep ocean's stable, slowly changing conditions can preserve entire body plans and evolutionary lineages that natural selection has long since eliminated from every shallower, more dynamically changing habitat on Earth. Beyond the goblin shark and its uniquely ancient lineage, the deep ocean supports an entire separate category of shark species, adapted to permanent cold and darkness, and few among them have proven as scientifically significant or as genuinely startling once properly measured as the Greenland shark, Somniosus microcephalus, a slow-moving, large-bodied species inhabiting the frigid waters of the North Atlantic and Arctic Ocean, generally at depths ranging from near the surface in the coldest polar waters down to more than 2,000 meters in warmer regions further south. For decades, marine biologists had no reliable method for determining a Greenland shark's actual age. The species entirely lacks the hard, ring-marked structures, such as otoliths in bony fish or growth bands in shark vertebrae, that scientists ordinarily use to count age, much the way tree rings reveal a tree's age, a genuinely frustrating gap given mounting circumstantial evidence, including extraordinarily slow observed growth rates of roughly 1 centimeter per year, suggesting Greenland sharks might live extraordinarily long lives. A breakthrough came through a genuinely creative research approach published in 2016 by a Danish-led research team headed by marine biologist Julius Nielsen, who realized that the lens of a Greenland shark's eye, unlike most other tissue in the body, forms in distinct during early development that remain once symbolically inert and essentially unchanged for the rest of the animal's life, preserving a chemical record datable through radiocarbon analysis. The same basic dating technique archaeologists use on ancient artifacts. Applying this technique to the eye lenses of 28 female Greenland sharks caught incidentally as fishing bycatch, Nielsen's team estimated the largest specimen studied, measuring just over 5 meters in length, to be approximately 392 years old, with a statistical range extending as high as 512 years, findings published in the prestigious journal Science that instantly established the Greenland shark as very likely the longest living vertebrate species confirmed anywhere on Earth, a title previously held by the bowhead whale, and one that implies individual. Greenland sharks alive today may have been born before large portions of recorded scientific history as we currently understand it had even occurred. This extraordinary longevity connects directly to an equally extraordinary reproductive timeline, since Nielsen's research team calculated, based on typical Greenland shark body length at sexual maturity compared against their newly established age-length growth curve, that female Greenland sharks likely do not reach reproductive maturity until somewhere around 150 years of age, meaning a shark born during the height of a historical era, like the Napoleonic Wars might, under this estimate, be only just reaching the point of being biologically capable of reproducing for the first time in the present day, an almost unfathomably slow reproductive pace that researchers studying the species have specifically flagged as a serious conservation concern, since a population capable of only extremely gradual generational renewal would recover exceptionally slowly over many human lifetimes from any significant population decline caused by fishing bycatch or environmental change. The Greenland shark carries a further genuinely distinctive biological trait connected directly to its deep, cold habitat. Its flesh contains unusually high concentrations of trimethylamine N-oxide, the same pressure-stabilizing compound discussed in connection with Haddle snailfish elsewhere. In this ocean story, a molecule that helps prevent the shark's own proteins from being crushed and deformed by the pressure differences it experiences moving between shallow Arctic water and considerably deeper dives, but that breaks down after death into trimethylamine, a compound with effects on the human nervous system similar to alcohol intoxication, if the meat is eaten fresh and unprepared, rendering fresh Greenland shark meat genuinely toxic and capable of producing a state Icelandic sources have historically described as shark drunk. Icelandic communities developed a specific traditional preparation method centuries ago to render the meat safely edible, called a carl, involving burying the shark's flesh in gravel and sand for several weeks to allow initial fermentation, then hanging the meat to air dry for several additional months, a process that breaks down the trimethylamine oxide sufficiently to make the final product safe to eat, still served today as a traditional Icelandic delicacy and offering a genuinely striking real-world illustration of how the exact same molecular adaptation that lets Greenland sharks survive centuries at crushing depth also happens to be the specific chemical reason eating the animal requires several months of deliberate preparation first. A separate deep-water species, the blunt-nosed six-gilled shark, Hexanchus griseus, offers researchers a genuinely useful anatomical throwback for an entirely different reason. Unlike the overwhelming majority of modern shark species, which possess 5 gill slits on each side of the head, six-gill sharks retain 6, an ancestral trait shared with several extinct shark lineages known only from the fossil record and generally understood by paleontologists to represent a more primitive condition than the five-gill arrangement that came to dominate most later shark evolution. Six-gill sharks range across an unusually wide depth span, with juveniles and adults documented anywhere from shallow coastal waters down to depths exceeding 2,000 meters, and researchers tracking tagged individuals using acoustic and satellite tags have documented a pronounced pattern of diel vertical migration remarkably similar in basic structure to the twilight zone migration. Many smaller fish and invertebrates undertake, with six-gill sharks generally remaining at greater depth during daylight hours and moving into considerably shallower water at night, a behavioral pattern researchers believe reflects the shark following its own preferred prey species as they undertake their own nightly migration upward toward the surface. Deep-sea sharks as a broader category present researchers with a genuinely difficult conservation puzzle, precisely because so many species across this group share the same basic biological pattern documented in exhaustive detail for the Greenland shark specifically. Slow growth, delayed sexual maturity, and low reproductive output, a life history strategy generally well suited to the deep-sea's stable, low mortality environment under natural conditions, but one that leaves these populations severely vulnerable to modern fishing pressure, since a population that might naturally lose only a small fraction of its members to predation or disease in any given year has essentially no capacity to compensate for additional mortality from bycatch or targeted deep-water fishing operations, meaning a deep-sea shark population, once significantly depleted, may require considerably longer to recover, if it ever fully does, than nearly any comparably sized fish population living in shallower, faster reproducing marine environments. Few large marine animals have been discovered as recently, or as unexpectedly, as the Megachasmopelagios, an entire previously unknown genus and species first encountered on November 15, 1976, when a U.S. Navy vessel operating off the coast of Hawaii accidentally snagged one in its sea anchor at a depth of about 165 meters, hauling up a 5-meter, filter-feeding shark unlike anything then catalogued in the scientific literature, distinctive enough in its anatomy that researchers formally classified it not merely as a new species, but as the founding member of an entirely new shark family, Megachasmidae, a genuinely rare taxonomic event for a large-bodied vertebrate discovered this recently in scientific history. Megamouth sharks feed, much like the considerably better-known whale shark and basking shark, by passively filtering plankton and small fish from the water using specialized comb-like gill rakers. But unlike those two more frequently encountered filter-feeding species, megamouths appear to spend the overwhelming majority of their time at meaningful depth, generally between 150 and 500 meters, only rising toward shallower water at night, apparently following the same diel vertical migration of plankton and small prey discussed in connection with several other deep-sea species throughout tonight's video. Despite nearly 5 decades of accumulated research attention since the species' accidental 1976 discovery, fewer than 300 confirmed megamouth shark encounters have been documented worldwide, as of the most recent published tallies, and researchers still lack basic information about the species' reproductive behavior, population size, and preferred breeding grounds. A genuine reminder that an entire family of large filter-feeding sharks, animals, measuring several meters in length, remained completely unknown to science within living memory, discovered not through any dedicated deep-sea expedition at all, but by pure chance, tangled in a piece of ordinary naval equipment. The deepest living shark species confirmed anywhere on Earth is the Portuguese dogfish, Centroschimnus coelalepis, a small, dark-bodied species reliably documented at depths exceeding 3,700 meters. with a small number of unconfirmed trawl records suggesting individuals may occasionally range even deeper still. Researchers studying Portuguese dogfish physiology have found the species relies on an unusually large oil-rich liver in some individuals, accounting for more than a quarter of the shark's total body weight, to help offset its own natural negative buoyancy at extreme depth, since sharks generally lack the gas-filled swim bladder bony fish use for buoyancy control, relying instead on lower-density liver oil and constant, energy-costly swimming to avoid sinking, a metabolic burden that becomes considerably more demanding the deeper, colder, and more food-scarce the surrounding water becomes. Portuguese dogfish additionally reproduce through a method called ovoviviparity, in which fertilized eggs develop and hatch entirely inside the mother's body before live young are eventually born, a reproductive strategy shared with several other deep-sea shark species discussed across tonight's video, and one generally understood by researchers as advantageous specifically in the deep sea's stable, predator-scarce environment, since it allows developing embryos to be carried and protected internally for an extended period rather than left vulnerable inside an external egg case on the open seafloor, though the specific gestation length for Portuguese dogfish, believed based on limited study to extend well over a year, remains only approximately established, given how rarely pregnant individuals are captured and studied in adequate detail at the depths where the species actually lives. Beyond the fish and cephalopods that tend to dominate popular imagination of deep-sea life, the ocean floor supports several genuinely unusual categories of organism that receive considerably less public attention despite representing some of the deep sea's most scientifically significant residents, beginning with a creature so strange that it wasn't formally identified as a distinct species until remarkably recently in scientific terms. The Yeti crab, Kiwa hirsuta, first collected and formally described in 2005 by a French-led research team exploring hydrothermal vent fields along the Pacific Antarctic ridge south of Easter Island, immediately drew widespread public attention for its genuinely distinctive appearance, dense, pale, hair-like bristles called setae covering its claws and forearms, giving the animal a shaggy, almost fur-covered appearance entirely unlike any previously known crab species, a resemblance striking enough that researchers formally named the entire new genus Kiwa after a mythological Polynesian goddess of shellfish, while the specific species name and its immediate popular nickname both drew directly on comparisons to the legendary Himalayan yeti. Detailed study of the setae covering a yeti crab's claws revealed something considerably more scientifically significant than simple camouflage or insulation. The bristles host dense colonies of chemosynthetic bacteria, the same basic, sulfur-oxidizing bacterial partnership that powers giant tubeworm nutrition at hydrothermal vents, and researchers studying yeti crab feeding behavior have documented individuals performing a distinctive, repeated waving motion with their claws, believed to actively circulate mineral-rich vent water across the bacterial colonies to maximize their growth, before periodically using specialized mouthparts to comb and consume the resulting bacterial growth directly from their own arms, effectively farming a food source on their own body in a behavior researchers have described as unlike anything documented in any other crab species studied to date. Considerably stranger still, and considerably less familiar even to people who follow ocean science closely, are xenophyophores, single-celled organisms belonging to a group of amoeba-like protists that construct elaborate external shells, called tests, out of sediment particles, mineral grains, and the discarded shells of smaller organisms cemented together with organic material the cell itself produces. Despite consisting of a single cell, xenophyophores can grow to genuinely enormous sizes by single-cell standards, with some documented specimens exceeding 20 centimeters in diameter, making them, by most working definitions, the largest known single-celled organisms on Earth. a title that regularly surprises people encountering the fact for the first time, given how thoroughly popular biology education associates single-celled life exclusively with the microscopic. A 2011 research expedition to the Mariana Trench, led by marine biologist Lisa Levin's team, documented dense concentrations of xenophyophores at depths exceeding 10,000 meters among the deepest points in the entire ocean. And researchers studying these deep populations have found xenophyophore tests frequently harbor a genuinely dense community of smaller organisms, including nematode worms, isopods, and juvenile fish, sheltering directly within the test's structural crevices, meaning a single, unassuming, sediment-colored blob on the seafloor may function as a miniature, self-contained apartment complex for an entire small community of other deep-sea animals. A genuinely significant ecological role for an organism most people have never once heard of. Deep-sea sponges present their own distinctive category of biological extreme, particularly the glass sponges or hexactinellids, which construct their entire skeletal structure from silica chemically identical to ordinary window glass, arranged into an intricate lattice of microscopic spicules. The Venus flower basket Euplectella aspergillum offers perhaps the most visually striking example, its skeleton forming a delicate cylindrical lattice cage so structurally elegant that materials scientists have directly studied its specific fiber optic-like glass architecture for potential engineering applications, and the species carries a genuinely well-known natural history. Curiosity. A small species of shrimp frequently takes up residence inside the sponge's central cavity as a juvenile, growing too large to ever leave once mature, spending the remainder of its life trapped, but safely sheltered, within the glass lattice, a relationship, traditionally referenced in Japan as a wedding gift symbol representing lifelong marital devotion, since the shrimp pair, once trapped together inside, remain permanently and inseparably bonded within their glass home. Antarctic glass sponges, studied through repeated photographic surveys of individually tagged specimens conducted across multiple decades by researchers including biologist Claudio Richter, grow at an extraordinarily slow rate in the frigid Southern Ocean, and comparative growth rate calculations have led some researchers to propose that the very largest documented individual sponges, based on their measured size relative to observed annual growth increments, could plausibly be several thousand years old, positioning certain individual glass sponges, if these age estimates hold up under further study, among the oldest living animals on the entire planet. Among the deep sea's less celebrated but genuinely striking giants are sea spiders or pycnogonids, marine arthropods only distantly related to true land spiders despite the superficial resemblance, which follow the same broad pattern of polar and deep-sea gigantism documented across numerous unrelated animal groups discussed elsewhere in tonight's video, with shallow-water tropical sea spiders typically measuring just a few millimeters across, while certain deep and polar Antarctic species grow, leg spans exceeding 70 centimeters, dramatically larger than any of their warm, shallow-water relatives. Researchers studying giant sea spider physiology have identified a genuinely puzzling biological workaround underlying their unusual body plan. Pycnogonids lack a conventional centralized respiratory or circulatory system entirely, instead relying on oxygen simply diffusing directly through their exceptionally thin exoskeleton and into extensions of their digestive tract that reach out into their unusually long, thin legs, a diffusion-based system that becomes progressively less efficient as body size increases under ordinary physical and chemical constraints, meaning researchers still don't have a fully settled explanation for precisely how the very largest documented polar sea spider species manage to sustain adequate oxygen supply to their extremities at the sizes they've been confirmed to reach, a genuinely open physiological question actively studied by polar biologists working the cold. distant edges of the world's least visited seas. Rounding out this chapter's catalog of overlooked giants, siphonophores, colonial organisms related to jellyfish and corals, but structured as a single connected colony of thousands of specialized, genetically identical individual units called zooids, each performing a distinct role, feeding, reproduction, propulsion, within the larger whole, produced what may be the longest documented animal on Earth when a 2020 Schmidt Ocean Institute expedition to Ningaloo Canyon off the coast of Western Australia filmed a single siphonophore, believed to belong to the genus Apolemia, estimated at approximately 45 meters in total length when its full, spiral-coiled feeding structure was accounted for, a length that, if the measurement holds up under continued scientific scrutiny, would exceed even the largest confirmed blue whale, making an organism most people have never heard of a serious candidate for the longest animal ever directly documented by science. Its entire body, remarkably, not one single individual organism in the ordinary biological sense at all, but a cooperative colony of thousands of genetically identical clones functioning together as one. Pyrosomes, another genuinely striking colonial organism related, somewhat surprisingly, more closely to vertebrates than to jellyfish despite their superficial resemblance to one, are built from thousands of small, individual filter-feeding animals called zooids, each embedded within a shared, tube-shaped gelatinous structure and cooperatively pumping water through the colony's hollow interior to feed and propel the entire assembly slowly through the water column, a coordination pattern that gives pyrosome colonies their common nickname, sea pickles, and their scientific name, which translates roughly to fire bodies, a direct reference to the vivid, synchronized bioluminescent glow the entire colony produces when disturbed. While individual pyrosome colonies typically measure only a few centimeters to perhaps a meter in length, researchers have documented rare specimens growing dramatically larger, including reports of colonies exceeding 20 meters, long enough that divers have on occasion mistaken a single colony for a translucent, glowing tunnel large enough to swim directly through. Pyrosomes experienced a genuinely dramatic and still only partially explained population boom along the west coast of North America beginning around 2014, when researchers and commercial fishing crews began reporting pyrosome numbers in some coastal waters increasing by what marine biologists studying the bloom described as several orders of magnitude compared to historical baseline levels, clogging fishing nets and washing ashore in unprecedented quantities from California to Alaska. A bloom oceanographers have tentatively linked to a period of unusually warm Pacific surface water occurring during the same general period, though the precise combination of factors that triggered and eventually caused the bloom to subside remains an active subject of ongoing research rather than a fully settled explanation. The Haddle Trenches discussed elsewhere in tonight's video, and in this channel's earlier deep ocean video, host their own dramatic case of deep-sea gigantism among an otherwise unassuming group. Amphipods, small shrimp-like scavenging crustaceans, typically measuring only a centimeter or two in length across most of the world's oceans, which grow to genuinely startling proportions in the deepest trenches on Earth. Elasella gigantea, first collected in the 1970s, but studied in far greater detail only through baited trap expeditions conducted over the past 2 decades, has been documented reaching lengths exceeding 28 centimeters, making it, by a considerable margin, the largest amphipod species ever recorded, more than 10 times the length of its more typical shallow water relatives. Researchers deploying baited camera traps at extreme Haddle depths, including a 2017 expedition to the Mariana Trench, have documented swarms of giant amphipods arriving and stripping bait carcasses within a matter of hours, evidence supporting the same general food scarcity hypothesis discussed earlier in tonight's video, as one explanation for deep-sea gigantism generally, since a body capable of consuming and storing an unusually large single meal offers a genuine survival advantage in an environment where the next opportunity to feed may not arrive again for a considerable stretch of time, and researchers studying trench amphipods have found the giant species to be genuinely widespread, documented across trenches separated by thousands of kilometers. Despite each individual trench functioning as an isolated deep-water basin, cut off from its neighbors by intervening shallower ocean floor. A distributional pattern whose precise underlying explanation, whether reflecting some form of larval dispersal through intermediate water depths or a genuinely ancient pre-trench formation distribution, remains only partially understood. Among the deep ocean's most visually and chemically counterintuitive features are brine pools, dense, hypersaline bodies of water that pool on the ocean floor and behave, in nearly every practical respect, like an entirely separate lake, complete with its own visible shoreline, its own surface, and even its own waves, despite sitting directly on the bottom of the open ocean, fully submerged beneath thousands of meters of ordinary seawater above it. The first scientifically documented deep-sea brine pool was identified in the Gulf of Mexico during the early 1980s, when researchers operating the submersible Alvin, the same vehicle responsible for the 1977 hydrothermal vent discovery discussed elsewhere in this ocean story, encountered a genuinely disorienting sight during a routine seafloor survey. What appeared through the submersible's viewport to be an ordinary lake, complete with a visible shoreline and a distinct, gently rippling surface, resting in a shallow depression on the seafloor more than 3,000 meters beneath the actual ocean surface far above. A discovery so visually counterintuitive that early researchers reportedly needed a moment to process what their own instruments and eyes were telling them simultaneously. The basic physics keeping a brine pool's water from simply mixing into the vastly larger body of ordinary seawater surrounding it comes down to density stratification, the same fundamental principle that keeps oil floating on top of vinegar in a kitchen bottle, because the brine's dramatically higher salt concentration makes it considerably denser than the ordinary seawater around it, and because the deep ocean floor at these depths sits in a state of near total stillness, with essentially no wave action or current strong enough to force physical mixing, the two fluids can remain in stable contact along a sharp boundary for years or even decades without ever substantially blending together, a genuinely rare, real-world demonstration of pure molecular diffusion operating with almost no interference from the turbulent mixing that governs how fluids normally behave almost everywhere else on the planet's surface. A separate and considerably larger Gulf of Mexico feature, the Orca Basin, discovered and mapped through sonar surveys conducted during the 1970s, and named for the distinctive orca-like pattern its outline traced across early sonar readouts, illustrates just how large a single brine pool can become, spanning roughly 400 square kilometers, making it, by total surface area, one of the largest known brine pools anywhere in the world's oceans, considerably larger than the more famous but far more compact Jacuzzi of Despair discussed above. Resting at a depth of approximately 2,400 meters and containing brine roughly 7 times saltier than ordinary seawater, the Orca Basin has drawn sustained scientific interest specifically because its stable, oxygen-depleted, highly saline layer traps and preserves organic sediment falling into it in a manner similar to the anoxic conditions documented in the Black Sea, creating a genuinely valuable, undisturbed sediment record that geologists have used to reconstruct regional climate and sea level history, stretching back tens of thousands of years. The basin's own extreme chemistry inadvertently functioning as a kind of natural, continuously operating archive of the Gulf of Mexico's own deep past. Brine pools form where underground salt deposits, typically remnants of ancient seas that evaporated, and left behind thick salt layers now buried beneath the seafloor, come into contact with fractures or seeps, and slowly dissolve into the surrounding groundwater, producing an extremely salty and therefore considerably denser fluid that flows out onto the ocean floor, and because it is markedly denser than the ordinary seawater surrounding it, settles into low-lying depressions rather than mixing and dispersing the way most fluids released into open water normally would. The result, filmed and directly explored by numerous submersible expeditions over the past several decades, is a genuinely striking underwater landscape, a pool of visibly distinct, denser fluid resting in a seafloor depression, complete with a sharply defined boundary, or pyknock line, separating it from the ordinary seawater above, one so physically distinct that researchers have documented submersibles capable of gently settling directly onto the brine pool's surface without sinking through it, and have filmed small waves genuinely rippling across that submerged surface, driven by the same basic density difference physics that keeps oil floating visibly on top of water, playing out in complete reversal several kilometers beneath the actual ocean surface far above. Particularly well-studied and evocatively named example, informally called the Jacuzzi of Despair, discovered in the Gulf of Mexico and studied extensively by researchers including biologist Eric Cordes, sits within a roughly 20-meter-wide circular brine pool, with salinity levels measured at more than 4 times that of ordinary seawater, water so dense and chemically hostile, combined with locally high concentrations of dissolved methane and hydrogen sulfide, that almost nothing entering the pool directly survives the exposure, and researchers documenting the site have filmed the genuinely grim spectacle the nickname directly references: mobile animals, including crabs and occasional larger fish, that wander into the pool's edge, apparently unable to detect the danger in time, becoming trapped, poisoned, and preserved by the brine's extreme chemistry, their remains accumulating in a visible ring around the pool's perimeter, a natural mortality trap operating continuously on the seafloor entirely independent of any human involvement. Despite the pool's interior proving lethal to nearly all mobile animal life that enters it directly, researchers have documented dense, thriving colonies of specialized mussels and tube worms growing directly along the brine pool's edge, adapted specifically to tolerate the extreme chemical gradient found at that exact boundary, harvesting energy from the same chemosynthetic bacterial partnerships that support life at hydrothermal vents and cold seeps discussed elsewhere in this ocean story, thriving in a narrow habitable margin, sitting directly beside a pool actively lethal to nearly everything else nearby. Beyond the Gulf of Mexico, the Mediterranean Sea hosts several deep hypersaline anoxic basins, considerably larger in scale than typical brine pools, and formed through a somewhat different geological process connected to the Mediterranean's own dramatic geological history, since the entire sea is understood, based on extensive geological evidence, to have nearly completely evaporated during a dramatic period called the Messinian Salinity Crisis, roughly 5.9 to 5.3 million years ago, when tectonic movement temporarily sealed off the connection to the Atlantic Ocean at the Strait of Gibraltar, leaving behind massive buried salt deposits across much of the modern seafloor that continue slowly dissolving into deep basin water even today. The largest of these Mediterranean basins, called l'Atalante, discovered and studied extensively by European research teams beginning in the late 1990s, was found by researchers including microbiologist Roberto Danavaro's team to be almost entirely devoid of dissolved oxygen and populated primarily by highly specialized microbial life adapted to the basin's extreme salt concentration, and a particularly striking 2010 discovery published by Danavaro's research group reported apparent evidence of tiny multicellular animals called loriciferans living and reproducing directly within the anoxic brine itself, a finding that, if fully confirmed through the additional independent verification, the original researchers themselves called for, would represent the first multicellular animal life ever documented capable of completing its entire life cycle in a permanently oxygen-free environment, a genuinely significant and still not entirely settled claim that continues to draw scientific attention and follow-up research given its considerable implications for how narrowly or broadly biologists should define the basic environmental requirements for complex animal life. The Red Sea hosts a genuinely distinct category of brine pool entirely, one defined by extreme heat rather than the cold, stable chemistry found in the Gulf of Mexico and Mediterranean examples described above. The Atlantis II Deep, discovered during oceanographic surveys in the 1960s, and sitting at a depth of roughly 2,000 meters within a tectonic rift zone where the Red Sea's own seafloor is actively spreading apart, contains brine measured at temperatures exceeding 60 degrees Celsius, dramatically hotter than the surrounding deep water and rich in dissolved metal sulfides, including commercially significant concentrations of zinc, copper, silver, and gold, precipitating out of the hot brine and settling as metal-rich mud on the basin floor beneath it. Researchers studying the Atlantis II Deep's origin have traced its chemistry to the same basic tectonic rifting process responsible for the Red Sea's ongoing formation as a young ocean basin with hot, mineral-laden fluid circulating through newly formed volcanic rock deep beneath the seafloor before emerging directly into the deep basin above, and the site's genuinely enormous estimated metal reserves, by some geological surveys among the largest known concentrations of seafloor metal sulfide deposits anywhere in the world, have made it a subject of serious, if still preliminary, commercial mining interest from Saudi Arabian and Sudanese government-affiliated ventures since the 1970s, a considerably longer and more geopolitically complex commercial history than the polymetallic nodule mining debate playing out elsewhere in the world's oceans. The deep ocean is, contrary to the near-silent image most people associate with darkness and isolation, a genuinely noisy environment filled with a constant background of biological, geological, and increasingly human-generated sound, and the U.S. National Oceanic and Atmospheric Administration has for decades maintained a network of permanently moored underwater microphones called hydrophones specifically to monitor and record it, a listening network that has occasionally picked up sounds genuinely strange enough to generate years of serious scientific mystery before finally being explained. The most famous of these recorded mysteries, informally nicknamed the bloop, was detected in the summer of 1997 by NOAA's Equatorial Pacific Ocean Hydrophone Array, an extremely powerful ultra-low frequency sound loud enough to be picked up by sensors more than 5,000 kilometers apart, a detection range that, based on how sound propagates through the ocean, initially suggested to the NOAA researchers analyzing it a source considerably larger and louder than any known biological animal, including the largest whale species, was capable of producing. The sound's genuinely unusual acoustic signature, its extreme volume combined with a frequency pattern that shared some superficial characteristics with sounds known to be produced by living marine animals, generated years of speculative public and even some cautious scientific discussion about what kind of creature, previously entirely unknown to science, might be capable of producing it. Speculation that spread widely once NOAA made an edited, sped-up version of the recording publicly available online. The mystery was formally and conclusively resolved in 2005 when NOAA researchers, having by then collected several additional years of comparable acoustic data from Antarctic regions equipped with similar hydrophone monitoring, determined that the bloop's specific acoustic signature closely matched sounds recorded from ice quakes, the sudden, large-scale fracturing of Antarctic ice shelves as massive sections calve away and crack under their own accumulated stress, a geological rather than biological origin that fully accounted for both the sound's enormous volume and its unusual, previously unmatched frequency profile, bringing a formal, satisfying, if considerably less exciting, end to one of ocean science's more genuinely entertaining recent mysteries. The bloop was far from the only unusual sound NOAA's Pacific Hydrophone Network detected during this same general period, and several other recordings, each informally nicknamed by the researchers who first identified them, generated their own smaller rounds of scientific investigation before eventually being resolved. A sound nicknamed Julia, detected in March 1999, and one nicknamed Slow Down, detected in May 1997, both showed acoustic signatures researchers eventually attributed, following the same general pattern established by the bloop's resolution, to a combination of iceberg movement, ice shelf fracturing, and, in Slow Down's specific case, the sound of a large iceberg dragging and grinding directly along the seafloor as ocean currents pushed it slowly across shallower coastal terrain, a genuinely dramatic geological process capable of generating sustained, powerful underwater sound entirely without any biological source involved at all. A further recording nicknamed Upsweep, first detected in August 1991, and notable for continuing to be recorded in a distinctly seasonal pattern for years afterward, remains even today only partially explained, with NOAA researchers' leading hypothesis pointing toward ongoing volcanic or hydrothermal activity at a specific, still not precisely pinpointed underwater location in the Pacific, though the sound's specific underlying mechanism and its curious seasonal timing pattern have never been as fully and conclusively resolved as the bloop's more dramatic mystery ultimately was. Whale vocalizations themselves contribute an enormous share of the deep ocean's total biological sound, and blue whales specifically produce some of the loudest sounds generated by any living animal on Earth, with certain calls measured at source volumes exceeding 180 decibels, a level that, while measured underwater using a different reference scale than sound measured in air, and therefore not directly comparable to familiar everyday sound levels, is nonetheless understood by researchers to be loud enough that, under the right conditions and propagating through the ocean's efficient deep sound channel, individual blue whale calls can potentially be detected by other whales, and by hydrophone arrays across thousands of kilometers of open ocean. Genuinely puzzling to researchers who have studied recorded blue whale calls across multiple decades is a well-documented, gradual, and remarkably consistent global trend. Blue whale call frequency has been steadily dropping, generation after generation, across every studied population worldwide, with a comprehensive analysis published in 2009 by researcher Mark MacDonald and colleagues, compiling decades of hydrophone recordings from multiple ocean basins, confirming the pattern held true independently across geographically separated and genetically distinct blue whale populations that have essentially no direct contact with one another, ruling out simple cultural transmission of a shared vocal trend as the explanation. Several competing hypotheses have been proposed for this consistent global frequency drop, including a connection to the partial recovery of blue whale population sizes following the end of large-scale commercial whaling, on the reasoning that whales calling across shorter average distances to more numerous near-individuals might not require quite as high or as far-carrying a frequency as whales calling across the more sparsely populated, whaling-depleted ocean of previous decades, though researchers studying the phenomenon are candid that no single explanation has yet achieved full scientific consensus, leaving the steady, synchronized deepening of the largest animal ever known to have lived on Earth's own voice a genuinely open acoustic mystery. While blue whale calls carry the title of loudest sustained sound, the single loudest sound produced by any biological source on Earth belongs instead to the sperm whale, whose echolocation clicks used to navigate and hunt for squid in the total darkness of the deep water they dive into, have been measured at source volumes, reaching as high as 230 decibels underwater, a figure that, once again measured on a scale specific to underwater acoustics rather than directly comparable to everyday sound experienced in air, is nonetheless understood by bioacousticians to represent the most intense sound any living organism is known to produce, generated by forcing air through a specialized structure in the whale's massive head called the spermaceti organ, which focuses the resulting click into a tightly directed acoustic beam aimed forward through the water like a biological sonar searchlight. Researchers studying sperm whale echolocation have found the clicks are powerful enough that some scientists have seriously proposed, though not yet conclusively demonstrated, that sperm whales might be capable of using an intensified burst of clicks to physically stun or disorient prey directly through acoustic force alone, a hypothesis sometimes called the Big Bang theory of sperm whale hunting, distinct from the animal's confirmed use of clicks purely for echolocation and prey detection, and one that remains a genuinely debated and only partially tested idea within the field of marine bioacoustics. Much of the hydrophone data researchers use to study whale calls, mystery sounds, and deep ocean acoustics generally now comes from a genuinely unexpected source, a global monitoring network originally built for an entirely different purpose. The Comprehensive Nuclear Test Ban Treaty Organization, established following the 1996 treaty banning nuclear weapons testing, maintains a permanent international network of underwater hydroacoustic monitoring stations positioned specifically to detect the distinctive acoustic signature of an underwater or near-surface nuclear detonation anywhere on the planet, exploiting the same efficient deep sound channel discussed elsewhere in tonight's video to achieve detection ranges of thousands of kilometers from just a small number of strategically positioned stations. Because the network continuously records and analyzes essentially all significant underwater sound within its detection range, not merely sounds matching a nuclear detonation's specific signature, marine scientists have increasingly gained access through formal data sharing agreements to this treaty monitoring network's incidentally collected acoustic archive, using it to track whale migration patterns across ocean basins, monitor underwater volcanic and seismic activity, and cross-reference historical mystery recordings, including several of the unusual sounds discussed earlier in this chapter, a genuinely unusual case of Cold War-era nuclear security infrastructure becoming, decades after its original purpose was established, one of marine bioacoustics' most valuable and geographically comprehensive research tools. Human-generated sound now contributes a genuinely significant and growing share of the deep ocean's total acoustic environment, and among the loudest and most sustained sources is seismic airgun surveying, technique the oil and gas exploration industry has used since the 1960s to map subsurface geology by firing powerful compressed air blasts into the water at regular intervals, sometimes every 10 seconds, continuously for weeks at a time, and recording how the resulting sound waves reflect back off different rock layers beneath the seafloor. Airgun arrays produce sound at source levels comparable to some of the loudest sounds discussed elsewhere in this chapter, and researchers studying the biological impact of sustained seismic surveying have documented measurable effects extending well beyond the immediate survey area, including a widely cited 2017 study published in Nature Ecology and Evolution, led by researcher Robert Macaulay, which found a single seismic airgun survey off the coast of Tasmania was associated with a measurable immediate decline in local zooplankton abundance, extending across a radius of more than a kilometer from the survey vessel, alongside significantly elevated mortality, specifically among krill larvae exposed to the sound, a finding with genuine implications given zooplankton's foundational role in ocean food webs including the twilight zone migration and biological pump processes discussed elsewhere across both of this channel's deep ocean videos. Marine mammal researchers have documented separate concerns regarding how sustained airgun noise may interfere with the same. Long-distance whale communication that exploits the deep sound channel, potentially masking calls or forcing whales to alter their own vocalization patterns to be heard over the artificial noise. And while the full scope of seismic survey impact on deep-sea ecosystems remains an active area of ongoing research rather than a fully settled scientific question, the mounting evidence has prompted several national regulatory bodies to impose new restrictions and mandatory mitigation measures on seismic survey operations in recent years, an evolving policy response to a form of ocean noise pollution most people have likely never once considered, despite it originating from a technology that has been continuously in commercial use for more than half a century. The deep ocean's combination of scarce food, stable temperature, and low overall mortality risk from predation has favored, across an extraordinarily wide range of unrelated deep-sea animal groups, an underlying life history strategy built around patience rather than speed, and few documented examples illustrate that strategy as vividly, or as record-breakingly, as a single deep-sea octopus tracked continuously by researchers off the coast of California. In May 2007, a research team from the Monterey Bay Aquarium Research Institute, using a remotely operated vehicle to survey a rocky outcrop at a depth of approximately 1,400 meters, discovered a female deep-sea octopus, Granelodon boreopacifica, guarding a clutch of eggs attached to the rock face, and rather than simply documenting the observation and moving on, the research team, led by biologist Bruce Robison, made the deliberate decision to return repeatedly to the exact same location over an extended period, specifically to track how long the brooding period would actually last, a commitment that ultimately produced one of the most remarkable endurance records ever documented in the entire animal kingdom. Over the course of 18 separate submersible dives conducted across 4.5 years, the research team found the same female octopus, identifiable through distinctive scarring, remaining in essentially the exact same position, continuously guarding and gently tending her eggs, and showing no evidence of feeding. During the entire observed period, her body visibly growing paler and more translucent as the years passed, and her own physical condition steadily deteriorated. The eggs finally hatched sometime between the team's final 2 dives, and the mother octopus, consistent with the semilparous reproductive pattern documented across most octopus species generally, in which an individual reproduces only once in its entire lifetime before dying shortly afterward, was gone by the team's final visit, her death presumably occurring shortly after her eggs successfully hatched, bringing her total confirmed brooding period to approximately 53 months, by a considerable margin, the longest egg brooding period ever documented for any animal species on Earth, a finding published in 2014 in the journal PLOS ONE that immediately became one of the most widely cited examples of extreme parental investment documented anywhere in the natural world. Researchers studying this remarkable case have proposed that Granelodon boreopacifica's extraordinarily extended brooding period likely reflects a direct trade-off shaped by the deep sea's near-freezing water temperature, since embryonic development in cold-blooded animals generally proceeds considerably more slowly at low temperature, and the extreme cold and correspondingly slow metabolic pace at the depth. This specific octopus was observed brooding her eggs would naturally extend the incubation period well beyond what a related octopus species living in warmer, shallower water would ever require, though the sheer multi-year scale of the observed brooding period still exceeded what most researchers studying deep-sea octopus biology had previously assumed possible for any cephalopod species, prompting renewed interest in studying reproductive timelines across other, less thoroughly documented, deep-sea octopus species. Chimeras, a lineage of cartilaginous fish commonly called ghost sharks or ratfish, despite belonging to their own entirely separate evolutionary branch that diverged from true sharks and rays somewhere around 400 million years ago, follow their own genuinely slow, deliberate reproductive pattern well suited to the deep continental slope waters, generally between 200 and 2,600 meters, where most species live out their lives. Female chimeras produce a small number of large, leathery egg capsules deposited directly onto the seafloor and left entirely unguarded by either parent, each containing enough stored nutrition to sustain the developing embryo through an incubation period that researchers studying captive and wild-monitored specimens have documented can stretch beyond a full year. In some species, an extraordinarily long incubation period for any fish made necessary by the slow embryonic development rates, typical of the cold, low-energy, deep-sea environment discussed throughout tonight's video. Chimeras additionally retain a genuinely distinctive anatomical feature found in no other living vertebrate group, a retractable sexual clasping organ on the male's forehead, in addition to the paired pelvic claspers shared with sharks and rays, whose specific functional role during mating researchers have only partially observed directly, given how rarely chimeras are ever encountered engaging in courtship or mating behavior at the depths where it actually occurs, leaving several basic details of chimera reproductive behavior inferred largely from anatomical study rather than direct field observation. Before the Greenland shark's own 2016 age confirmation displaced it, the longevity record among vertebrates belonged to the bowhead whale, Balaena mysticetus, a large Arctic baleen whale species whose extraordinary lifespan came to scientific attention through a genuinely unusual line of physical evidence: traditional ivory and stone harpoon points, weapon styles that Alaskan indigenous whaling communities had stopped manufacturing and using by the early 20th century in favor of more modern equipment, were found still embedded in the blubber of bowhead whales, harvested during subsistence hunts conducted decade. Later, direct physical proof that specific individual whales had survived an attempted hunt using equipment retired from active use generations earlier. Following up on these striking individual cases, researchers led by biologist Jeffrey Bada applied a chemical dating technique, measuring the gradual racemization, or structural conversion, of specific amino acids within the whale's eye lens, a slow chemical process that occurs at a generally predictable rate over an individual's lifetime, and their analysis, published in 1999, estimated several examined bowhead whales at more than 200 years old, with the oldest specimen studied estimated at approximately 211 years, findings that established the bowhead as easily the longest-lived mammal species confirmed anywhere on Earth, and prompted renewed scientific interest in what cellular and genetic mechanisms might allow such an enormous, long-lived animal to apparently avoid the elevated cancer risk that cellular aging theory would otherwise predict for a creature with so many more cells and so many more decades of accumulated cell division than almost any other mammal alive. This extended pattern of slow growth and delayed, energy-intensive reproduction extends well beyond octopuses and the deep-sea sharks discussed earlier in tonight's video, and researchers studying deep-sea bivalves, particularly giant clams belonging to the genus Calyptogena found at cold seeps and hydrothermal vents, have documented individual specimens estimated, based on shell growth ring analysis, to live for well over a century, growing at rates dramatically slower than related shallow-water clam species that can reach full maturity within just a few years. A particularly striking demonstration of just how differently deep-sea and shallow-water growth rates can diverge comes from direct comparative studies of ocean quahog clams, Arctica islandica, a species found across a considerable depth range from relatively shallow coastal shelf waters down into deeper offshore habitat, in which Researchers have documented that individuals living in the coldest, deepest portions of the species range consistently show slower growth and correspondingly greater maximum lifespan than genetically similar individuals living in warmer, shallower water nearby, with the single oldest confirmed ocean quahog specimen ever recorded, a 2006 catch off the coast of Iceland informally nicknamed Ming, by the research team that later determined its age through shell growth ring counting, estimated at 507 years old at the time of its collection, meaning the animal had been alive since before the founding of the Ming Dynasty era. Print the researchers referenced when choosing its nickname, a specimen whose extraordinary age was only fully confirmed after the animal had already been inadvertently killed during the very research process used to study it. The orange ruffy, Hoplostethus atlanticus, a deep-water fish found around seamounts at depths between roughly 500 and 1,800 meters, offers a genuinely sobering case study in exactly what happens when this same slow-growth, delayed maturity strategy collide directly with modern industrial fishing rather than simply being studied from a safe scientific distance. Orange ruffy were essentially unknown to commercial fisheries until the late 1970s, when trawling fleets began targeting the large, easily located spawning aggregations. The species forms around specific eastmounts, and the fishery expanded rapidly through the 1980s based on the mistaken assumption, common at the time, that a deep-sea fish this abundant in aggregated catches must reproduce at a pace broadly comparable to more familiar, shallower water commercial fish species. Subsequent research, including age analysis using the same otolith growth ring counting technique applied to numerous other fish species, revealed the orange ruffy's true biology only after considerable damage had already been done. Individual orange ruffy can live to at least 150 years old, with some specimens estimated at over 200, and the species does not reach sexual maturity until somewhere between 20 and 40 years of age, a life history profile radically unlike the fast-growing, quickly maturing shallow-water fish species most commercial fishery management models had originally been built around. Several major orange ruffy fisheries, most notably around New Zealand and Australia, subsequently collapsed dramatically during the 1990s and 2000s, once the true extent of overfishing became apparent, and the species has since become one of the most frequently cited case studies in fisheries science for the specific danger of applying fast fish population models to a slow-growing deep-sea species, a cautionary example that directly informed the more conservative management approaches now applied to other newly discovered deep-sea fisheries, including several of the seamount and slope fish populations discussed elsewhere across both of this channel's deep ocean videos. Even the most remote and physically inaccessible parts of the deep ocean are not, researchers have found through decades of increasingly precise long-term monitoring, chemically isolated from what happens at the surface and in the atmosphere far above, and tracking exactly how quickly and how deeply human-driven atmospheric change is actually reaching into the ocean's deepest waters. It's become one of oceanography's most significant and technically demanding ongoing research efforts. Ocean acidification, the gradual, measurable decrease in seawater pH driven by the ocean's absorption of atmospheric carbon dioxide, a gas that reacts with seawater to form carbonic acid, has been documented most extensively in surface water, where direct atmospheric contact makes the chemical change easiest to measure and track. But researchers studying deep water chemistry have found the effect penetrating to genuinely significant depth as well, carried downward by the same thermohaline circulation system that continuously cycles cold, dense water from the surface into the deep ocean over long timescales. A particularly significant and genuinely concerning consequence for deep-sea ecosystems involves the same carbonate compensation depth discussed earlier in tonight's video, the boundary below which calcium carbonate shells dissolve faster than they accumulate. since multiple published studies tracking long-term ocean chemistry data have found this boundary has been measurably shifting to shallower depths in several ocean regions as deep water absorbs more dissolved carbon dioxide and becomes correspondingly more acidic, meaning organisms that build calcium carbonate shells or skeletons, including many of the cold water corals discussed elsewhere in this ocean story, may face measurably more corrosive conditions at depths where they have successfully thrived for thousands of years, a genuinely significant long-term threat. Researchers are still working to fully quantify across different ocean regions and depth ranges. Directly monitoring these slow-moving deep ocean changes with adequate precision required an entirely new generation of oceanographic instrument, and the Deep Argo program, an expansion of the broader, well-established Argo float network that has continuously monitored the upper 2,000 meters of the global ocean since the early 2000s, was specifically developed to extend this same automated monitoring capability down to the full ocean depth of 6,000 meters. Deep Argo floats, battery-powered autonomous instruments that drift with deep ocean currents while periodically descending and ascending to record detailed temperature, salinity, and pressure data before transmitting their findings back to researchers via satellite. Whenever they briefly surface, began large-scale deployment during the 2010s, and the resulting long-term dataset has allowed oceanographers, for the first time in the field's history, to directly and continuously track subtle but genuinely significant warming trends occurring in the deep ocean itself, rather than relying on the comparatively sparse, occasional research ship-based deep water sampling that had constituted the overwhelming majority of deep ocean temperature data collected throughout the 20th century. That deep ocean warming trend, once properly measured through sustained deep Argo monitoring, has proven scientifically significant specifically because of how much heat the deep ocean is now understood to be quietly absorbing on humanity's behalf. Multiple published analyses of the global ocean heat budget, compiling data from Deep Argo floats alongside other deep ocean temperature monitoring programs, have found that the ocean as a whole has absorbed more than 90% of the excess heat trapped by human-caused greenhouse gas emissions since the mid-20th century. And while the majority of that absorbed heat remains concentrated in the upper, more thoroughly studied layers of the ocean, researchers tracking Deep Argo data have documented measurable, if considerably smaller in magnitude, warming trends extending into the deep ocean as well, particularly in specific deep water formation regions near Antarctica, where the same cold, dense water that sinks and spreads slowly across the entire global seafloor originates, meaning changes occurring at the surface in the coldest, most remote regions of the planet are, through this same slow deep water circulation system, gradually reaching essentially the entire global deep ocean, a process researchers estimate could take centuries to fully play out, given how slowly deep ocean circulation moves, but one whose earliest measurable stages, Deep Argo, technology has only become capable of directly detecting within roughly the past decade. A further and genuinely significant deep ocean chemistry concern involves oxygen minimum zones, naturally occurring layers of the ocean, typically found at intermediate depths, roughly between 200 and 1,000 meters in specific regions, including the eastern tropical Pacific and the northern Indian Ocean, where dissolved oxygen levels drop dramatically due to a combination of high biological productivity near the surface, which consumes oxygen as decaying organic material sinks and decomposes, and comparatively sluggish water circulation that fails to adequately resupply the depleted oxygen from elsewhere. Researchers compiling multiple decades of oxygen measurement data from these specific regions have documented that oxygen minimum zones have been measurably expanding, both in total geographic area and in vertical thickness across several major ocean basins since systematic monitoring began, a trend generally attributed to a combination of warming surface water which holds less dissolved oxygen and also increases the density stratification that limits vertical mixing, and potential changes in large-scale ocean circulation patterns, and marine biologists, studying the ecological consequences, have found that expanding oxygen minimum zones are measurably compressing the habitable vertical range available to numerous fish and invertebrate species that require higher oxygen concentrations than these expanding low oxygen layers can provide, effectively squeezing certain populations into a narrower band of habitable water, sandwiched between adequate, surface layer above, and the expanding, oxygen-depleted zone pressing upward from below. The broader deep water circulation system responsible for distributing both heat and oxygen across the global ocean, the thermohaline circulation discussed earlier in tonight's video, has itself become the subject of genuine scientific concern in recent years, specifically regarding its Atlantic component, formerly called the Atlantic Meridional Overturning Circulation, or AMOC, the specific current system responsible for carrying warm surface water northward through the Atlantic before it cools, sinks, and returns southward as cold, deep water. Multiple independent research teams analyzing distinct lines of evidence including deep ocean temperature and salinity records, direct current strength monitoring arrays maintained since 2004, and indirect reconstructions drawn from ocean sediment cores, have published findings over the past decade, suggesting the AMOC has weakened measurably compared to earlier in the 20th century, with a widely discussed 2018 study led by researcher Lev Caesar proposing the system may be at its weakest point in more than 1,000 years based on temperature pattern evidence in the North Atlantic. The specific mechanism of concern involves melting Greenland ice sheet freshwater, diluting the salty, dense surface water that normally sinks to drive the entire circulation system. Since freshwater is less dense and therefore less prone to sinking, and while the AMOC has shown meaningful natural variability across its recorded history, making any single study's conclusions about a long-term weakening trend genuinely debated within the broader oceanographic community, the possibility that this specific deep-water circulation system, the same basic process responsible for distributing cold, oxygen-rich water across the entire ocean floor over the course of roughly a 1,000-year global circuit, could slow significantly or, in the most extreme and still highly uncertain modeled scenarios, potentially destabilize entirely within the coming centuries, has become one of the more actively monitored and genuinely consequential open questions in contemporary climate and ocean science. The same biological pump process that carries carbon downward through the twilight zone's nightly migration has inspired a genuinely controversial proposed climate intervention called ocean iron fertilization, based on the observation that phytoplankton growth across large stretches of the open ocean, particularly in the Southern Ocean, is limited primarily by the availability of dissolved iron rather than by sunlight or other nutrients, a finding first proposed by oceanographer John Martin in 1988 and tested through more than a dozen small-scale research experiments conducted since the 1990s, in which research teams deliberately dispersed dissolved iron across patches of open ocean and measured the resulting phytoplankton bloom. Proponents of large-scale iron fertilization have argued the technique could, in principle, be deployed at a meaningfully large scale specifically to trigger phytoplankton blooms large enough to draw down significant additional atmospheric carbon dioxide, with a portion of that carbon eventually sinking to the deep sea floor as the resulting biological material dies and settles, functioning as a deliberate engineered enhancement of the same biological pump process already occurring naturally. The approach has drawn substantial scientific skepticism and, in several cases, formal international regulatory restriction, since research teams studying the small-scale experiments conducted so far have found the actual proportion of resulting biological carbon that reaches and remains permanently sequestered on the deep seafloor rather than simply being consumed and re-released by other organisms within the food web before ever sinking that far, appears considerably smaller and less predictable than early proponents originally hoped, and the London Convention and London Protocol, international treaties governing ocean dumping, were formally amended in 2013 specifically to restrict large-scale ocean fertilization experiments, pending considerably more rigorous scientific evidence of both their genuine carbon sequestration effectiveness and their broader ecological safety, leaving iron fertilization a technique that remains scientifically intriguing in principle, but that most current ocean policy treats as unproven and genuinely risky to deploy at any meaningful scale. We've spent tonight moving through 10 chapters and 180 facts entirely distinct from the deep ocean's more familiar catalog, from abyssal hills, the single most common landform on the entire planet, through a Victorian scientist's confidently wrong theory that life stopped at 550 meters, into a fish presumed extinct for 66 million years pulled from a fishing boat's net, past sharks that may remember the era of the Napoleonic Wars, through crabs that farm bacteria on their own arms and single cells the size of a fist into underwater lakes with their own shorelines and waves, past a mysterious sound that spent 8 years unexplained before turning out to be Antarctic ice, through an octopus that gave nearly 5 years of her life to a single clutch of eggs, and finally into the slow, patient, decades-long work of measuring exactly how far the surface world's changes are reaching into the deep. It's worth sitting in these last quiet minutes with how much of tonight's material, like the 150 facts covered in this channel's earlier deep ocean video, comes from a field that is still, by any honest measure, in its early stages. The coelacanth wasn't rediscovered until 1938. The Greenland shark's true age wasn't confirmed until 2016. The longest documented animal on Earth wasn't filmed until 2020. Deep Argo floats have only been watching the deep ocean's slow temperature shifts for about a decade, measured against the age of the ocean itself, which has existed in something close to its current form for well over 4 billion years. Essentially, everything humanity currently understands about its deepest waters has been learned within the span of a single, very recent, very short human lifetime. There's a particular comfort available in sitting with that mismatch for a moment, rather than rushing past it. Edward Forbes was wrong about where life stopped, confidently and publicly wrong, and the ocean simply kept doing what it had always done, regardless of what any single scientist, however respected, had concluded about it from the deck of a ship. The stalked crinoids didn't know they'd been declared nearly extinct. The coelacanth wasn't hiding from anyone. It was simply living exactly where it had always lived, entirely unconcerned with what paleontologists a continent away happened to believe about its continued existence. The deep ocean has never once needed human confirmation to keep being exactly what it already was, and it isn't waiting on any of us now either. That gap between the ocean's age and our own knowledge of it was never really the discouraging part of tonight's journey, if you sit with it for a moment before letting the thought go entirely, the way you might set down something heavy you've been carrying without quite noticing. The Greenland sharks, currently alive right now, some of them older than the country you may be listening from, are not waiting on anyone's discovery to keep circling slowly through cold Arctic water. The abyssal hills have been quietly rolling across the seafloor, entirely unmapped and entirely unbothered by that fact, since long before anything resembling a human being existed to wonder about them at all. The brine pools have kept their shorelines steady in total darkness for longer than any coastline on dry land has looked the way it currently does. Whatever remains unknown down there will still be unknown tomorrow, and largely unknown for a good while after that, patient in a way nothing on the surface ever really has the luxury of being. Somewhere right now, a Greenland shark, older than most nations currently in existence, is moving slowly through black Arctic water in no particular hurry, the way it has been moving for longer than anyone alive today has been alive at all, and will likely still be moving that same unhurried way long after tonight's listener has gone. A nautilus is beginning its own nightly rise toward a shallower reef, following a route its ancestors were already following before there were mammals large enough to notice, let alone curious enough to ask why. None of it is waiting on tonight's video, or on any video ever made, to keep happening exactly as it already has for longer than the whole of recorded human history has existed to write any of it down. So let this be the last current pulling at you tonight. Let the deep ocean keep doing what it's done for longer than any of us can properly picture, holding its slow-growing clams and its shrieking icebergs and its shaggy bacteria-farming crabs, in the same total, patient darkness it always has, entirely unconcerned with whether anyone is listening, and let everything you've heard tonight settle now, gently, the way marine snow settles, slowly and patiently, and without any hurry at all the way down into sleep.